Hey, good afternoon. Boa tarde. Today we have with us here in this webinar Dr. Ricardo Hayashi. And for me, it's an honor to have this guy here. He is my former student. I think one of the first students that I have when I started as a professor. So very welcome, Ricardo. Thank you so much for your time.
Thank you, Elizabeth. It's such a big pleasure to be here talking about this topic with you, and it's gonna be nice. Thank you for the invite.
Yeah, so we remind everybody that stay listening us this afternoon that you are able to do questions, just to write your questions here and we will answer it. And at some time, if it's not possible during this period, we can do later on. So please be our guest to let your questions here for Dr. Hayashi. So, Ricardo, when you start, I know when you start because I was there. Of course, that the age it was not different because I was so young, but still, of course, I'm just kidding. Ricardo, would like to tell the people what's your experience or when you start to study microbiota and how this start to influence in your career as a scientist, as a professional.
Yes. So thank you for this time. It was not that long, right, Elizabeth? We both of us are very young. It was not that long time ago.
I always do.
Yes, so I was your student, you were my advisor at the time. I started to work with intestinal health in general with you and our research group in 2006, I guess. And since then, it was very like we studied a little bit more about the gut health in general, histopathology, immune responses with some challenges. So we did a great job doing this kind of work at that time. So I had my master's degree and my PhD from the same university, UFPR, Federal University of Paraná State in south of Brazil, Curitiba. And during my PhD, I went to University of Illinois, located at Urbana-Champaign, and worked with almost 2 years with Dr. Roderick Mackey. So there, I had the opportunity to work a little bit more about this topic. We had this great chance to work with some researchers from Michigan State University, and we went to Bermuda Island to study a little bit more about feral chickens. Just to explain a little bit more about feral chickens is a group of chickens that has some genetic traces from the modern broiler chicken, but this group of individuals, they went back to the wild environment. So they become like feral animals, but they have like some domesticated genetic traces. So it was very nice opportunity to work with this topic because it was something different at the time that I used to do it, because it was a project some with some exploration, some evolutionary stuff, which is very nice to understand, have some insights about what we are doing here in the industry. And this group of feral chickens show us a little bit some differences with the modern broiler chicks, because we
Yes.
did this comparison.
Before you talk about the difference, I would just try to put together the scenario because I remember when you told me that I want to go um, to Illinois, and I remember that you are so excited because this professor he really does not work exactly with animal production. And I remember that you were very excited because he said, "Oh, he works with many different species. He works also with babies and everything." And then later on, when you have this opportunity, I remember that I read on Science magazine about this feral chicken. So this group of the university uh, find these birds that was actually the regular birds, but went back to the wild, and you are very excited about that. So I think that this idea that microbiota as the first invited that I have here in this seminar, in this webinar series. We talk about the importance of the cross information between species, right? I think that the beautiful in this microbiota aspect is that we really can connect the information of different species, also plants, also environment, soil, and everything. So, what was the results that you got from these birds that you went to Bermuda? It was not bad, right? It was not
Yeah,
a bad
it was
experience
not
to
that
be in
bad
Bermuda.
because no, it was not that bad because once we were doing the experiment, we were looking for the beautiful beaches there, so it was very nice. But exactly, Elizabeth, because I decided to go with Dr. Mackey because he is a peer microbiologist, and sometimes we need to stay out of the box, think a little bit more about the whole scenario, have insights from other species. He works a lot with human infants, for example. So I had the opportunity to bring some insights to animal production, specifically for poultry. So it was very nice. And in Bermuda, when we started this project, we observed something very different because we compared both gut microbiomes from these feral chickens from the modern broiler chickens that we used to work, right? And then we saw a big diversity, a huge, bigger diversity from gut microbiome for this group of fairy chickens compared to the modern ones. So for us, it's very nice because we observed also a core microbiota, some common bacteria, some communities that we observed some sharing of this information and bacteria from both species, but also we observed some colonies, some specific communities that we have never seen before in the modern broiler chickens. So we hypothesized something that maybe we are losing some communities, some important communities of bacteria, other microorganisms that could help us to improve productivity, weight gain, feed conversion, and animal welfare as well. So for me, it was very nice because we need to think about what we are doing right now. If we are going to the right way, we need to think about the whole population, the interaction with the environment, and everything. Not only control some pathogens, which is very important. I know that. But it's very important to see the whole scenario, the whole picture, not only some specific bacteria, some specific biomarkers.
Yeah, in going for these directions, I would like to mention here Dr. Billy Argus because I really love the presentation when he said that, for example, it's a little bit out of what we are talking here, but just to bring some information that is more practical, maybe for people that is there. It's like he said that coccidiosis, for example, is a disease that we promote in the animals, like because in nature we don't have coccidiosis. However, we have the myria there, right? So what you're saying could be also related to because you use all the diet, like a regular diet with the ingredients that is normally the same, like corn, soybean. Of course, they have different differences in between, but they don't have so much diversity in what they eat, right? And now we start new ideas. For example, the insect meal, different kind of ingredients that we are forced to use because we need to use a different kind. So how could we apply this information, Ricardo? Because I know you have been my student, you have been students of another professors and learn a lot, and now you are in one of the biggest company that produces animal broilers specifically in this case. And how do you apply this knowledge that you have from the science point of view in your day by day work at the company that you work?
Yes. So 2 years ago, I had this great opportunity to join the BRF Corporative Animal Health team. Today, I'm in charge of animal health innovation, so I need to be focused on what's going on out there about the new technologies, how to bring this new innovation, new stuff to the industry. So this topic, Elizabeth, not because I have been studying this subject for a long time, But it's for me, it's very important to bring this concept of gut microbiota, because if we have this package of analysis, we're gonna have time, we're gonna have condition, we're gonna have data, information to make some decisions, right? This is very important for me. And you told about the coccidiosis, for example, right? Because it didn't exist in another scenarios outside of animal production, for example. It's very for me it's a kind of consequence that we are doing, because the system that we are producing right now, it's very controlled. We have a lot of biosecurity procedures, very strict disinfection procedures, which is going to modulate somehow the microbiota of whole environment, not only the gut. So you're going to select somehow some groups of bacteria, some group of microorganisms. This is good for one reason, because you can control some pathogens like salmonella, some clostridium, listeria, for example. But in the other way, it's gonna be like a negative result as well, because we're gonna have some susceptibility to have more infections about other microorganisms, and you're gonna select bacteria, and through a long time you're gonna lose some specific groups, depending on how do you do your procedures and all the other stuff that you do in animal production.
Okay, in the practical way, Ricardo, how do you guys apply this information? Is just a research now, or do you have like some apply actions that you guys are doing with this information about microbiota?
It's not just for research, because we are doing this project with a lot of partners right now. When I say partners, I say like startups, universities, and other companies that are helping us to sequence the samples and try to understand a little bit more. But before this step, Elizabeth, it's very important to have like a powerful question. What do we need? What do we need to know about this data, about profiles, about phylogenetic trees, classifications? Because this is the first step. What do I want to do with these questions, right? And after this, it's very important to have like a standard of analysis of the gut microbiome, because it's very complex. There's a lot of information, and we need to focus on how to interpret these results. So we need methods, right? It's not just research. It's research, but we are trying to apply for our demands, because we have some questions, some demands from the field that we are trying to solve them. And I think this method, if we do in the right way, is going to help us to gain some time, to have time for interventions, predict some diseases, right? And compare some scenarios and have like a specific R&D tool to make decisions in the animal industry.
So there is any kind of tool that you guys are using? Like, do you standardize procedures you analyze in the same lab? How is like the practical way for you to, or you're just now collecting information?
We are collecting information right now, Elizabeth, but we are very like standardize the patterns, the communities, how to do this sampling from everything. So this is very important before you start your just sampling, right? We need to do the powerful question, for example. We need to correlate, for example, some communities with good performance or bad performance. This is the first step for me, because we need to correlate with everything that we already have, right? And after that, we need to answer some second questions like, can we correlate these microbiota profiles with biosecurity procedures? Or maybe if we put some feed additive, can we modulate this? And well, to summarize everything, we need to have like big data. We need to sampling, we need to put in the platform, and then who knows, in the future we're gonna create like an algorithm and work with artificial intelligence to have these interpretations. And for us, it's gonna be nice and huge to make decisions on this data.
Yeah, I think this is amazing because even the IC inside method, when people talk about, they think it's about histology or something, but the concept, it was right like this, like you need to put all the information together and understand it. Do you think you have enough tool to do it? Like we really are prepared in terms of, I mean, when I say we, I mean our animal industry it's prepared to put all together these informations, or we need to do some adjustment for have all these informations together in a way that we can really make a decision from it.
Yes, this project, like I said before, it's very complex. So it's not possible to do it by just ourselves, right? So we have to find the right partners to do it. When I say partners, I say people from statistical people from technology, bioinformatics, right? People from the field, nutritionists. So I think it's very complex, and we need to put the people all together to do this project. This is the hardest part, because it's a very like complex to put the knowledge in just one project and execute it. So this is one important part that we are doing right now.
As you know, last week I was here with Dr. Furquet, and we talk a little bit about the situations, because you are a company, and I know that every company in the world today, everyone has a lot of compliance things, and sometimes when a big project like that, it's like at some point you have to put a lot of interests together, like you are doing it in your company, you say you have a partners, but do you think that this is could be also drive or share with another company like you, or is something that you are just designed for your situation for BRF? This is the question.
Well, I think we are doing just by ourselves right now, but I really believe that to make a standard of this package of analysis involving gut microbiome should be like global. You know what I mean? Because we need to have like a big data, something worldwide to share the information. Because, for example, if we're gonna study like the gut microbiome of poultry, this is gonna be very different scenarios, different methods, so we need to have like the big data and try to compare with this database. You know, for now we are doing just by ourselves, doing with the right partners, but we need to have like a specific worldwide bank of the informations to share and compare. You know, maybe we can train our algorithm or something like that using artificial intelligence with this common big database, right?
Yeah, we are here today, like of course each one of us here, or even people that is not in front of cameras now, but that listen us. And once again, you can ask a question all the time. So if you are uh, willing to have a questions, please do it. We are here, and we send it to Ricardo. But what I'm saying is, like, of course, that we are here representing some companies, but at the end of the day, like as a scientist that I am, and I know that you are as well, I really dream to have this platform to be global. Like someone take this and could be like the one that put all the informations from all the companies together, because I think when you have a communion of all this information, doesn't matter at the end. And people are afraid sometimes when they talk about big data. I have been here also with Aidan, and we talk about that people are afraid to give the results, because they think that we're going to share the results. But it's important, Ricardo, to tell a little bit that we when we receive these informations, but we don't know where they come from. This is possible, so we are just having all the informations together. And when I start to talk about that some years ago, I always use like the Facebook as a way to say, like, people are very interested that people are talking about you in the Facebook, but you are not really you. All the algorithm are coming as something that has your profile but not need to be you. Can you talk a little bit about this, how the big data could be uh, used to this purpose that we are talking here?
Yes, it's a very nice question, because we already are doing some specific projects not focused on gut microbiota right now, involving big data in BIF. So we have like a platform to predict some situations that we need to pass through the situations. So the results from this kind of projects was very nice, because we are now working with a strategic way, and that I really need to we need to think about the whole picture and share the information with all the scenarios, different companies, not only in Brazil but Latin American and other
Yes.
continents, because we need to train this algorithm. You know what I mean? Because the more data that you have, the better quality you're gonna have in the result. You know, so for depends on the question, depends on the projects, but we are experiencing lots of projects inside of BIF predicting some scenarios, right? And I think very soon we're gonna put this information of gut microbiota that we are sampling in the field right now, and we're gonna put all together, and we're gonna have like a huge numbers, good data, good facts to make decisions. It's gonna be very nice.
You know, I'm very curious here if there is any information that you can share with us that you already have. That something that you have. Of course, you need to put more things in the algorithm, but something that you already have as informations about what you already saw in your big data analysis.
It's I cannot say like very details about this the
Oh, yeah, of
projects
course.
involving predictions, right? But it's very nice because we have the information, Elizabeth. You know, I think all the companies have lots of
Yes. Yes.
informations about the zootechnical parameters, the results, the past, the carcass yield, the situations, the procedures. So it's a huge big data, but we didn't know how to do this with this all the information. So for this project, we are putting all together. We train some algorithm from specific questions, and we are having like a good accuracy of this data in the results. So we can predict some scenarios from diseases, some situations that we are passing through, but at that time it's too late. When it happens, it's too late, right? We need to be like make like an anticipation, have time to do another way, and not to have like losses in the field. I cannot say a little
Yes.
bit details about these projects, but the most the main concept about this is kind of a common for artificial intelligence. You can apply for diseases, you can apply for um, uh, weight gain, for example, scenarios from zootechnical parameters. You can apply for gut microbiota. You can put the gut microbiota in the middle of this and try to predict some scenario. This is going to be very nice. So it's a huge world involve big data. It's very exciting and very needed to put in this in the animal industry.
Yeah, I'm also think that Hayashi is gonna we start with microbiota to talk about microbiota here and how to apply and everything. But I may also think that for the sustainability of animal productions, we need to go for it because, for example, let's put some not from your data. Let's put from general information that we have. We had here is the second time that we receive um, um, the professional from BRF who have a very nice conversation with your colleague Marco Price here. And when you talk with him, he used a lot of the concept of reuse the litter. For me, this is the most applied microbiota concept that we learn from the field. Like we reuse the litter and you have a better performance that when I start, I will not say the year, but when I start, that we learn that I remember Professor Alexandre Rosa saying to me, Santa Maria, we always need to put not because he was wrong, it's just because this was the concept at those times. He said, you need to put the chickens in a very beautiful, completely clean, and warm litter for them to be growing and have a good performance. And today we see it in different way. Can you comment a little bit about because this is already a well known. It's not specific from your company. However, I know that you guys have. A lot of knowledge that can you comment about this applied situation of the microbiota in the field using the reuse of litter?
Yes, we know from I don't know the last 10 years that putting like reusing the leader like a biosecurity procedure like cleaning and disinfection, it's very important to have the good results. It's very proved. It's already improved, been proved from the last 10 years, for example. And now we can see some very nice works, recent published papers showing this relationship about the leader with the gut microbiome. It's a kind of they both of systems can share the same community somehow. So I don't know, maybe I don't know in the future we have like a biomarker from the leader instead of doing something specific from the gut. Because it's very similar, of course the diversity is very different, but they can share some ecosystem, very core core ecosystem from both situations. For example, Faecalibacterium prausnitzii, which is a very good bacteria, very correlated with good performances. When you reuse the leader from different periods, a long period we use the leader, you're gonna have like the increase of this bacteria in the gut as well, in the cica of the birds. So we have a lot of markers that are showing us that the more you use the leader, the more better results you're gonna have because you're gonna modulate for good the environment all around the chickens, right? And one
Oh,
nice
this information.
example, Elizabeth,
Yeah, just go ahead. Yeah.
is an example that we are seeing some specific published papers about the human beings, for example. Babies that that were born in by C-section, for example, they have more susceptibility to have like problems with respiratory tract, diabetes, obesity, and something a lot of problems in the future, just because of the type of the birth as well. So maybe
Yeah,
we can do this connection about how clean the environment should be, or maybe we can modulate for good to have more contact, to stimulate the immune responses, and to have this modulation as soon as possible, to have better results and better animal health as well.
yes, maybe. Do you think that maybe in the future we can think that we have like a core or signature of microorganisms in the litter that you're gonna analyze in a very fast, quick kit, and say, oh, with this I can use reuse this litter. This is well done, and then we can predict the weight gain and the performance based
Yeah,
on the litter that we start the check.
I really believe that because the facts are there. The connection between the liver and the gut, the cecal microbiota, it's very, very nice, and we can have a lot of insights. I think in the near future, we can translate the concept of gut microbiome for the liver as well. And maybe the liver could be like a very indicator to predict some scenarios, some diseases as well. So I think we are very close to there.
Yes, and many countries, especially here in Canada, we are not allowed to reuse the litter. You know, it's not all the world that has the same concept that we have in Brazil. US also have the same concept. Europe is very resistant to that. So this difference between the countries, different regions in the planet, we're gonna change so much when these informations come over and we have more to do. And do you think, Hayash, that we're gonna change the vaccination? We're gonna change the nutrition based on it as well, or how do you feel it?
Well, I think when we put this kind of package of analysis, we can have more data about the gut microbiome. We can do everything. We have the chance to test all the possibilities. We can change the ingredients. We can change the concentration of each thing that we are putting in the feed. We can change the environment. We can change procedures, and then we're gonna have insights of what's going on, what is working or what is not. You know what I mean? So I think we got to be more precise about all the situations, all the questions that we had before.
And how long is it gonna take, Ricardo? We are talking about. Well, I hope I can be still smile in between this happens. How long you gonna think that you gonna start to apply it in more realistic way in your animal production?
I think there's no it's a very brand near future, Elizabeth, because we already do this. We're already doing this in the company.
Like
We
5
have
years, 10 years, 15
no
years?
less than this. I think in a couple of years we're gonna have like very consistent results about
Wow.
field conditions and correlations with everything that we are talking about right now. But one problem, Elizabeth, because today we are passing through a phase that 95% of the analysis is mostly by phylogenetic tree, the composition and the
Hmm.
quantification of these groups. For me, one big step to evolve about this whole package of analysis is to go deeper a little bit more and trying to analyze the function of all bacteria, all the communities. This is something that we are just seeing in universities and something very strict scenarios. And I think this is going to take a little longer to bring this concept and try to do the omics in general, to put together with this phylogenetic tree and have more information. I think this, when this happens, I think it's going to be complete and very nice information. For now, I think just the limitation is just for phylogenetic tree, which we can get great results about the behavior of these communities, the quantification of these groups. But I think in the near future, when we have all the omics together in this package, I think we're gonna have like very consistent about the function of the gut microbiome in general.
Well, we have a lot of questions here. We have from Vanice. It was nice to see to know that you are here too, Vanice. A big hug. And she asked the same, like, what is the practical way? We have Patricia here also asking, Patricia Rocha. She asked us, asked you, in fact, if you are looking for microbiota profile by age, by model of productions, ABF, conventional, and if there is some correlation of microbiome with zootechnical results. So of course, people want more deep, but you can just comment
Yes,
me.
I think I told you before about the powerful question. We need to do the powerful questions at the beginning. We need to know what we are doing there, what kind of information do we need to know, right? So we are trying to apply for different situations, but I think the first one is try to correlate with good and bad performances. After that, we can play with this kind of designs in the field. We can measure different systems, for example, non-antibiotics ever with AGP-free systems. How can we compare both systems? For example, we can compare like genetic lineages. If we're putting like two different genetic in the field, maybe the host can modulate differently the gut microbiome. I don't know, right? So
Mm-hmm,
this is the
mm-hmm.
powerful question. We can apply for everything, Elizabeth. We can apply for nutritional subjects. We can compare scenarios. So I think it's very multi-diverse. And about the age that Patricia asked, is we are trying to apply this package as well from very the initial phase, because we have seen some chronic mortality in the field that we cannot explain why it's happening, because there's a lot of factors, and I think it's a good way to understand a little bit more about this demand from the field. But I think until from the very beginning, after the hatchery until 20 days, I think is the best way to measure and see differences from modulation of gut microbiome. After that, it's very the gut microbiome starts to stay like stable, and it's very hard to see the
Black.
differences.
Uh-huh.
So it's very nice to see from the hatchery until 20 days of age. It's a good time to have this data, to have this information in comparison areas.
So to establish the good microbiota is the key, like you're saying. And when you talk about this methodology, are you selecting a specific? Can you talk a little bit about how do you select the to sampling this microbiota, and what standard that you take care during the sampling that you already see difference?
Yes, mostly we should be very careful about the sampling, because we need to design a very nice experiment from the field, because in the field we're going to have different factors that could affect the design, right? But the methodology, mostly when I say about sequencing, is mostly a phylogenetic tree. But basically what we are doing now, it's the composition and quantification of these families, genera, big groups, right? And then we can put in the algorithm, for example, and try to correlate with the results of that specific flock. So we need to sampling a lot, like a whole year, to have enough information and try to correlate, oh, this group is increasing a lot. Not only one bacteria, right, Elizabeth? Because I really think that one bacteria does not have like a specific effect to the performance. I think it's a very complex scenario. So big groups, I think it's we need to see the behavior of different groups and scenarios.
I'm very interested really to see if there is a group of microorganisms that affect it. But I'm also wondering here, Ricardo, about the metabolites, because like Mike always said, you can have like a different microorganisms, but doesn't mean that you have different metabolites or different results because you have different. So, are in your project also something else, or is microbiota that you really focus right now, and how do you think about the other
Yes.
parameters?
I think the metabolomics will be very complementary. It's going to be very complete, the project, but right now we don't have like specific tools to apply the metabolomics to this analysis. Like I said before, mostly of what we are doing now is the phylogenetic tree. It's 16S mostly, right? Sequencing and try to have this correlation with groups, specific groups, to feed conversion, weight gain, and specific scenarios. So mostly right now it's sequencing, but in the near future, I think proteomics, metabolomics, and transcriptomics should be aggregated to this analysis, this package of analysis, and we're gonna have better results than we're having right now.
So, do you think that you when you talk about this analysis that you guys are doing, um, we are talking about all the company, or you guys choose a percentage that will be studied first? How do you guys design? Because the company is big, right? So the project will start. Do you guys start as a pilot, or you guys already went for everything?
Yes, we put some specific questions in the very beginning. We are trying to compare some scenarios, and we are trying to apply this for different micro regions, because we do know that in south of Brazil, for example, we have like a similar structure, a similar type of production. But if we go to the Middle West, for example, it's totally different. It's a new structure, different types of productions somehow, and we need to get data from everything. So we have some pilots in some specific areas to collect these differences, and then we're gonna put all together to train this algorithm if we're gonna have in certain way.
So when you talk about sampling, you said that the intestine and the litter, they are very similar. So what you guys are focused now in litter, in intestine? What is the mainly sampling for you today?
Mostly cloacal swabs, Elizabeth, right now. And for just one project, we are trying to have this correlation with litter with some specific procedures, but mostly 95%, mostly making like information of gut microbiome.
Okay, directly from the animal. We have some questions here about the reuse litter. For example, Bruna Belotti asked for how many times is necessary to reuse the litter to have like a stable microbiota in the litter. Do
Yes,
you
that's a
have
good
this?
question. After 3 or 4 cycles of the flux in the same bed, we're gonna have like a stable gut microbiome. But it's very important to say that depends on the procedures that we are doing in the downtime, for example. It's not just put the leader there and try and see the flux going on. It's very important to have like very good procedures in the downtime, to guarantee the good microbiomes and try to modulate this litter to the best way, and we're going to see the good performance of the animals.
You guys are doing it at home, like all the microbiota analysis are doing it in your own laboratory, or you have partners for that?
Yeah, no, we have partners for that. We just do the sampling, and we have partners from bioinformatics, people from statistics, people inside of BRF as well are helping us to interpret these results. But mostly the lab part of this, the hard part of this analysis is doing outside of BIF with some partners that are sequencing and doing the bioinformatic analysis for us.
Wow, this is a very interesting project, Ricardo. I'm very, very happy that you are sharing some information with us, and also very happy that you are here. It's always nice to see how much for me, when I saw one of my students doing very well, and most of them are doing very well, it's like it's time for me to go back and just watch it. For me, it's so amazing to see it. So we are almost at the end of our time here. Time passed very fast. And we have some questions. I'm sorry, we are not able to answer all the questions now, but later on I will send it to Ricardo and he can answer you. But for us to close all this information, Ricardo, I'd like that you give a final message for how the people about what is the future for microbiota applied science, let's say.
Well, I think the first step, Elizabeth, is to understand and standardize this analysis. Because right now, when we see studies, published papers, they have different methodologies, different ways to see, to interpret these results. So somehow it's very confusing for everybody who is reading, who is getting the first contact with this topic. I think the first thing to do, we talked about this in the poultry science a couple weeks ago. To maybe we can create a group, a specific group of specialists about gut microbiome, and try to do this big data information. Try to put there and try to standardize and make like a guide for everything for everybody who wants to apply this methodology in the field or in universities. And well, I know in every kind of situations. So I think the first step is try to standardize this because we need to put all the knowledge in the same level and try to get the most of the results because somehow we are seeing some papers doing, oh, if we put this probiotic we're going to up regulate the lactobacillus and down regulate the clostridium. For me it doesn't mean anything at all because we need to see the whole picture. The ecosystem of the gut microbiota is very complex, is very huge. So we need to put the methodology and scientific method to interpret this. So for me, it's very challenging to bring these concepts from the industry, but it's possible, right? If we work with the right partners, if we work with good people trying to understand about this topic, which is a bridge from everything, it's a possibility to work with biologists and nutritionists and people from statistics, informatics. So it's very nice project because we can explore everything that we have the most, the biggest and the better, the most, to one project and try to understand and help us to improve the animal productivity, right?
Very nice, Ricardo. Thank you. Thank you so much for your time. We are in the end here. So thank you everybody that stay with us. Next week we talk about swine microbiota and we talk with Professor Chambeau from the University of Manitoba from here, Canada. So thank you so much, Ricardo, once again. Thank you
Thank you,
and
Elizabeth.
I see you
Such a
soon
big pleasure
next
to be
week.
here.
Okay, bye bye.
Bye bye.